Good afternoon, everyone. Good afternoon, everyone. Thanks for coming. My pleasure to be here opening this meeting in which we're going to talk about molecular control of mitochondrial metabolism and the other issues related to mitochondria. It's really such a nice topic to be discussed today, and I hope you enjoy, guys. First of all, I have some notes. I'd like to express my thanks to FeSBE Organizing Committee, in special Professor Hernandes Faustino Carvalho and Professor Ana Paula Davel for invitation and also for all support to get this meeting held. It's also important to bring up here that the meeting, you're going to have 5 different talks of 20 minutes. And on the end of each talk, you're going to have 5 additional minutes for questions or even some comments. Well, I think that's it. And let's get down to the first speaker. My pleasure to invite André Oliveira. Dr. Oliveira works at Obesity and Comorbidities Research Center under my supervision. He has academic background in biology with expertise in biochemistry and physiology. And he's going to talk about the role of PABPC4 and NCoR1 in skeletal muscle cell under stressing condition. And I would say that he has a very warm finds to show us. So André, thank you. And please tell us a little bit more about your finds. Thanks a lot.
Thanks, Professor Leonardo. I will share my screen with you. Could you see my presentation?
Yeah.
So,
okay.
Good afternoon, everybody. It's a pleasure to share some of my results here, the results I got during my PhD, my postdoc, under supervision of Professor Leonardo, as he said previously. And the title of this presentation is Concerted Action of NCoR1 and PABPC4 in response to metabolic stress. So we will give you some background, just a few minutes. It's quite simple. So it's well known that metabolic syndrome is one of the major health issues at the present, and metabolic syndrome is a set of metabolic abnormalities such as insulin resistance, diabetes type 2 diabetes, obesity, dyslipidemia, hypertension. And if we take a look at the number of patients with type 2 diabetes, for example, it's clear that it's an important issue, and new strategies must be employed to face this. And one of these strategies is to search for druggable molecules that could be modulated by drugs, and disrupted protein-to-protein interaction, for example. And an interesting approach is to look at the proteins that are related to oxidative metabolism and mitochondrial biogenesis, since metabolic regulation is largely dependent on mitochondrial function. Mitochondrial biogenesis is influenced by environmental stress, such as exercise, caloric restriction, low temperature, oxidative stress, cell division, renewal, and differentiation. And all this process requires a coordinated response to keep the regulation and the maintenance of this homeostasis is achieved mainly by transcriptional regulation. The transcriptional regulation can be fine-tuned by means of co-regulators, and co-regulators can be divided into co-activators or co-repressors. One of the most known and by far most studied co-activator is PGC1-alpha, which binds to NCoR, to PPAR nuclear receptor, for example, driving the gene expression of genes related to fatty acids, metabolism, or electron transport chain subunits, for example. On the other hand, one of the most studied co-repressor is NCoR1, and the nuclear receptor co-repressor, NCoR1. In a broad sense, when a co-repressor is bound to a transcription factor or nuclear receptor, it keeps the histones in a tightly packed, because the co-repressor complex has some enzymes which are responsible to remove acidyl groups from histones. And when an activating signal reaches the cell, for example, when a hormone binds to its receptor, it triggers a signaling pathway that leads to a conformational change in the nuclear receptor, which lose affinity with co-repressor and recruit co-activators, leading to gene transcription. So the aim of this study was to identify new proteins that might regulate mitochondrial biogenesis or metabolism. And to do that, we decided to use NCoR1 as a bait and perform immunoprecipitation assay to identify new proteins that was bound to NCoR1. We send the samples immunoprecipitated with NCoR to mass spectrometry to identify these proteins. And besides the classical NCoR1 interactors, we also found two new proteins, PABC4 and PFKM. I call it PABC4 only PAB, just from now on, just for a matter of simplicity. So C4 stands for Polya binding protein complex. As its name says, it is a protein which binds to the Polya tail in mRNA molecules, stabilizing them and increasing the protein population rate once it keeps the mRNA proteins away from degradation. And PFKM is pyruvate fructokinase. It's a glycolytic enzyme. And as we are more interested in proteins related to gene transcription and gene regulation, we decide to stay studying PAB4. So after we identified this protein, we validated our mass spec result, performing a reverse IP, which I overexpress this protein, PAB4, and blotted again plotted against NCoR1. As you can see here in control, there is no interaction, but in the cells I overexpress before there is still interaction with NCoR1. So indicating that the interaction really happens in cell is not an artifact. As this protein is related to mRNA stabilization and binding to mRNA, we decide to rule out the possibility that interaction between NCoR and PAB4 was mediated by RNA. So we treated the sample, the cell lysates, with RNAs. As we can see here, we still have the interaction between them. So we concluded that PAB4 and NCoR1 in fact interact, and this interaction is not mediated by RNA. And so after that, we start thinking about how this protein level, protein content is in models known to be to have a mitochondrial function during mitochondrial gain and function. For example, during muscle cell differentiation. In this example, we can see the differentiation process of C2, C12 cells, that is a muscle cell, a mice muscle cell, and during this process of cell differentiation, there is a shift from glycolytic to oxidative phenotype in these cells. And this is due because the cells, the number of mitochondria in cells is increased. And also, we analyzed the protein content during this differentiation process, and the protein content was really reduced. Another model well established and related with mitochondrial biogenesis is exercise training. And in this mice subjected to 12 weeks of training, we also see a clear reduction in PAB4 content compared to sedentary mice. And when we look at the other results from other groups, we also see that the mRNA is decreasing in microarray experiments during muscle cell differentiation, and also after exercise training. So this gives us some hints that this protein could be related with mitochondrial function or mitochondrial biogenesis process. So we decided to knock down this protein in this gene in the muscle cell, these 2C12 cells. And the knockdown was about 80% and 90% both at the mRNA or protein content. And when we analyze some mitochondrial parameters, such as oxygen consumption, we see a clear increase of mitochondrial function in this PAB4 knockdown cells. There is an increase at basal respiration ATP linked even maximal or spare capacity is increased during this due to this PAB4 knockdown. Other parameter accessed was the mitochondrial mass, which was performed to use MitoTracker as a specific tie to mitochondria. And we also confirmed that increase in mitochondrial number, mass, I'm sorry. And when we look at the lactate production and release into the media, there's a reduction in knockdown cells. On the other way, the citrate synthase activity was increased. Also, the number of mitochondrial DNA, the copy number was increased. So there is a clear increase in the oxidative phenotype due to PAB4 knockdown. We also look at these parameters in mast cells to analyze other model, not in a muscle cell. And also there is an increase in knockdown cells. Also, here I show you the effect of low glucose in the culture medium, which is known to increase oxygen consumption, and is also a model to show decrease in NCoR activity. Glucose is a stimuli to degrade. Low glucose is a stimuli to degrade NCoR. So we also analyzed the mitochondrial mass. As previously, there is an increase in mitochondrial mass due to PAB4 knockdown, and also the lactate release into the media was to the culture media was reduced. As according in the same fashion as the experiments, the results obtained using muscle cells. So we updated that model from the beginning. I show you the coordinated action of co-regulators to fine-tune the transcription, but now adding PAB4 as an interactor of NCoR1, and based in our results, and to keep studying this effect on NCoR activity, repressing the transcription activity of transcription factors. We perform a reporter assay to assess the effect of knockdown of PAB4 in the PPAR response element. So this is the sequence in the chromatin which PPAR binds to and leads to an increase in gene transcription of its target genes. So as I said before, it's well established in the literature that low glucose increase the activity of PPAR by itself, mostly because one of the cause is because NCoR is degraded, it's more degraded in this situation. And also we use another treatment, which is GW, that is non-PPAR activator. So in both treatment, we can see an increase in the response element, the activity of the nuclear receptor. And when we look at the knockdown of PAB4, we see that knockdown is enough to increase the activity of PPAR on its responsive element. And the other condition is increased even more when compared to control cells. We look at we performed the other experiment using the same approach, which is the reporter assay to PPAR. And we saw when we knock down the PAB4, we have a clear reduction in the activity of PPAR. But when we overexpress NCoR as expected, there's a reduction in the activity of receptor. And the PAB4 knockdown kindly reduce the repression caused by NCoR1 overexpression. We also accessed the mRNA levels of some electron transport protein genes, and there is a tendency to increase in knockdowns in PAB4 knockdown cells, but it's a clear increase when I look at the PCS1L, which is a mitochondrial chaperone important to complex tree assembly. We also see there is a clear reduction, a clear increase in complex 2 protein content when we knock down this protein. So the possible explanation to this reduction could be one might know that by reduction in mRNA and NCoR transcription, gene transcription, because PAB4 is a protein related to mRNA stabilization, but that's not the case, as there is no change in mRNA levels or gene expression of NCoR in PAB4 knockdown cells. And the other thing we considered there was the protein, NCoR1 protein content was decreased once the mRNA was not changing during PAB4 knockdown. And in fact, there's a reduction in NCoR content during caused by PAB4 knockdown. So at this moment, we are investigating this relationship between this correlation between PAB4 knockdown and NCoR protein content. So updating that model, and then I show in the beginning of presentation, we have NCoR directing with transcription factor inhibiting the gene transcription, and we found in our experiments that PAB4 interacts with NCoR1, and the PAB4 knockdown is enough to lead to increasing in PPAR transcription activity. And the possible mechanism is because NCoR1 is being more degraded by ubiquitin proteasome pathway. It's well established that NCoR1 is degraded by this pathway, but we are trying at this moment, at the present time, to understand if this pathway is really impacted by PAB4 knockdown. So what we have at the moment is that PAB4 is it's important to decrease NCoR levels and facilitating or decreasing the repression caused by NCoR1. So the conclusions we can draw at this moment of the project is PAB4 interacts with NCoR1 in a non-RNA dependent manner. PAB4 protein content is decreasing during stress condition in the same fashion as NCoR levels, and PAB4 knockdown increase mitochondrial oxidative phenotype. So I want to thank Professor Leonardo Silveira for all support and supervision. All members from Professor Leonardo, former members, Tanus, Igor, and the present members, Dimitrios, Bianca, Laís, Marcos, Patrick, Ninon, and Mariana, and now members from OCRC and Source Research Center. I also would like to want to thank the financial support provided by FAPESP, CAPS, and CNPq, and thanks for your time.
Thank you, André. Wonderful talk. Now we are open to questions.
Can I stop my video to share my screen?
You can keep it. I think so.
Okay.
Questions?
Leo, I believe you have one question in the chat.
Okay. Question in the chat. Very nice presentation. Is there any relationship between PABPC4 and PGC1 alpha expression?
We have not performed this experiment, but to be honest, I don't know. We don't focus in PGC1 alpha because it's very well studied, and NCoR is I wouldn't say less important, but by the contrary, but it's not as well studied as PGC1 alpha. So I wouldn't say if there is a relationship between these two proteins. It's an interesting question.
Thank you, Heraldo, for a question. Tanus, what happens when you overexpress PABPC4?
Not much, Tanus. We don't have a reversal of the effects. I tried, but there is no. I couldn't see a rescue of the phenotype.
Thank you.
Really good question.
André, I have a question out of your focus. When you make your immunoprecipitation, you took together with PABPC4, PFK, right?
Right.
Do you believe that under stress condition, it's well known that you have low levels of NCoR1 under this condition, you can regulate the glycolysis, increasing the activity of PFK?
This could be we have talked about that, and it's an interesting question. We also consider to take a look at this protein because it makes sense to decrease when we decrease when you change the glycolysis activity, during an stress, for example. It could be another node of regulation, the pathway. I don't know. It's interesting.
Mm-hmm. Okay, thank you.
I really appreciate all the comments, and thanks for your time.
Thank you, André. And now let's move on to the second speaker. My pleasure to invite Angela Saito. She's a research member of the National Laboratory of Bioscience Campinas, that you call as LNBio Campinas. She has a very good CV, as you can see through the list of high quality papers published. And today she's going to talk about cellular and mitochondrial metabolism and knockin animals models of human disease. So Angela, thanks a lot and feel free to get your talk started.
Hello, everybody. Can you hear me?
Yes.
Okay, so I'll share my presentation. Can you see my presentation?
Yeah, you can see, Angela. It's all right.
So, first of all, I'd like to thank Professor Leonardo and Neurons for inviting me for this presentation. It's a really pleasure to present my work in this symposium. And today, the title of my presentation is Investigating Cellular and Mitochondrial Metabolism in Animal Models of Human Disease. I'm a researcher at the Brazilian Bioscience National Laboratory, which is located in Brazilian Center for Research in Energy and Materials in CNPE Campinas. Today I will, my presentation will have results of a project that started in collaboration with Dr. Ana Kropisky and Carla Rosenberg from University of São Paulo. They have access to patients with intellectual disability and their exome sequencing, which is a technique for sequencing of all protein-coded genes in the genome. And after this exome sequencing, they obtain a list of genes and mutations that some of them may be related with the disease. So that performed in vitro structural and functional characterizations for one of these mutations. And then we created a mouse model for this mutation. So today, the focus of my presentation will be the mouse model, which we explore the aspects of brain cytoarchitecture, neurophysiology, mouse behavior, cellular and molecular.
Angela, sorry, there is a lot of noise during your presentation.
Let me see.
Okay. Can you try again? Yeah, now it's okay. It's work out.
Okay.
You can move on. Okay. Thank you.
Okay, thank you. So today the focus of my presentation will be the results with this mouse model, in which we focus in aspects, we study aspects of brain architecture, neurophysiology, behavior, and cellular and molecular studies in this mouse model. The protein that we've been studying is UBE2A, which is ubiquitin conjugating enzyme E2A from the ubiquitin signaling pathway. And this is this E2 enzyme, which catalyzes the transfer of ubiquitin from E1 enzyme to E3 ligase or directly to the protein substrate. And this post-translational modification regulated several biological processes that can go from proteasome degradation, endocytosis, DNA repair, and so on. Mutations in this enzyme can lead to a disease called X-linked intellectual disability type nascimento, which is actually a syndrome that has symptoms that go from moderate to severe intellectual disability, often comorbid with other neurological and behavioral features, such as epilepsy, autism, and developmental delay. And other mutations are already known. Other mutations in UBE2A are already known and are found in patients with intellectual disability. This is a representation of some of the missense mutations. And the missense mutation that we've been studying is this one localized close to the
Angela, sorry again. Some people are saying that the sound is very, very slow. Can you become it louder, please?
Okay. Now it's better. Can you hear me better?
Yeah, it's much better now. Thank you.
Okay. So the focus of our mutation, our mutation is localized here in close to catalytic site of UBE2A, and I hide this, the name of this mutation, this presentation, because we haven't published the results of the animal model yet. But as I mentioned before, we've published the structural and the functional characterization of this mutation, and we found that the mutation affects the ability of this enzyme to form a polyubiquitin chain. And this inhibition could be recovered by increasing the pH of the reaction. So we decided to create a mouse model for this mutation in UBE2A, and we used the CRISPR-Cas9 technology for that, and microinjection into mouse zygote. This is a picture of the microinjection and a picture of zygote during the injection. And you create a knockin animal model for this missense mutation in UBE2A. This enzyme, this is the structure of the gene, and for the knockin strategy, we use a single guide RNA targeting the 5th exon of the gene, as well as Cas9 endonuclease and single-stranded oligodeoxynucleotide. So Cas9 was used to induce a DNA double-strand break, and the single-stranded oligodeoxynucleotide was used to have homologous recombination and precisely insert the specific mutation. So for confirm the introduction of this mutation, we performed PCR amplification, followed by restriction enzyme digestion and DNA sequencing. So we went to investigate the brain of this mouse model, and here we have the results of Golgi staining of cortex and hippocampus brain tissues that we performed to have access to dendritic spine morphology and the densities. And we found that mutant dendritic spines have more immature morphology. I mean, they were thinner and shorter, while when we count the number of spines, I mean the density. We couldn't see difference in the density, but in the morphologies of these spines. Then we went to investigate the ultrastructure of the synapses by transmission electron microscopy. And here you can see images of cortex and hippocampus. And you can see the presynaptic terminal with several synaptic vesicles, the active zone and the postsynaptic bottom. When we counted the number of synaptic vesicles, we observed a decrease in the number of these synaptic vesicles in the mutant tissue. Together, these results indicate that our mouse model have impairments in brain cytoarchitecture and points to alterations in synaptic communication. So we went to study this synaptic function by using primary hippocampal neurons and fluorescent probes for analysis of calcium signaling, synaptic vesicle cycle, and mitochondrial calcium uptake, all of them with confocal microscopy. And these results are results with fluor4-AM probe, which is a cell-permeant calcium indicator. In this first graph, neurons were stimulated with glutamate, which binds to glutamate receptor, leading to neuron depolarization and an increase in calcium, intracellular calcium levels, as you can see here. And we observed that mutant neurons harbor shortened calcium peak amplitude compared to the wild type. When we stimulated these neurons with potassium chloride, we couldn't see this difference. So these results suggest that these impairments should be related to glutamate receptor and excitatory synapse transmission. We also performed synaptic vesicle recycling measurements in neurons, and for that we used FM143 dye, which stains synaptic membranes. And we observed that mutant neurons have more spontaneous exocytosis compared to wild type. And this difference was not observed after potassium chloride stimulation. We also performed the mitochondrial calcium uptake measurement by using rod2-AM probe. And we observed that in mutant neurons, mitochondria could capture more calcium in mutant neurons than in wild type neurons. So because increases in mitochondrial calcium concentration can lead to mitochondrial dysfunction, we went to investigate deeper mitochondria in our mouse model. So these are images from transmission electron microscopy of cortex and hippocampus brain regions. And we observed that while in wild type animals, mitochondria have this tubular, normal organized crystal. In mutant tissue, crystals were swollen and more disorganized. We also measured the area of all of these mitochondria and observed an increase in the area of mitochondria in the mutant tissue. So to have more insights related to brain metabolism and mitochondrial dysfunctions, we went to measure the ATP levels by HPLC, and we observed that in hippocampus, we have a decrease in the ATP concentration. We also observed an increase in mitochondrial superoxide, which is a ROS, reactive oxygen species. And all of these results point to mitochondrial functional and structural impairments in our mutant model. We also performed behavioral tests with this mice, and this one is called Barnes maze, which is used to investigate alterations in spatial learning and memory. In this test, mice are put on a round table with poles in the periphery, and during 4 training days, by using these colored cues in the room, they learn how to find the target hole, which have this escape black box here. So after each trial, in each training day, the search strategy was categorized and in random, serial, or special strategies. And we observed that our mutants mice learning how to find this escape box, but they performed poorly in aspects related to strategy of search, as we can see in this E and F panel. They took more time to learn the spatial strategy and search strategy. We also performed the other behavioral tests, and by using open field maze and elevated plus maze, we found that our mutant model have more anxiety-related behavior. So this test explores the natural preference of mice to dark spaces and avoidance to illuminated and open areas. And in here we can see that our mutant mice spend less time in the center of this open field maze and less time in the open areas. So so we conclude that our mutant mice have more anxiety-related behavior. And finally, we performed Metabolism to have more insights about the metabolism of this animal. And we found in brain tissues and in serum an increase in purine metabolism. We found an increase in inosine and hypoxanthine in brain, and an increase in adenosine in serum. And the literature discussed that increased free adenosine indicates cell damage or metabolic stress, which may support our findings of synaptic and mitochondrial impairments in this work. In conclusion, I showed you that we create a mouse model for a human disease, and we showed that this model has several impairments related to brain architecture, neurophysiology, and behavior, and we showed some mitochondrial and cellular metabolism alterations in this mouse model. I'd like to thank our collaborators at NBio, University of São Paulo, and UNICAMP, as well our financial support and CNPM, and your attention.
Thank you, Angela. Now we are open to questions.
I have a question. So very nice work, very nice presentation, Angela. Really cool. And I was wondering about the ubiquitination part. Since this enzyme is a ubiquitin enzyme, you have, it seems that you have accumulation of dysfunctional mitochondria. So I was wondering if this accumulation of dysfunctional mitochondria could be associated with the loss of mitophagy by the ubiquitin pathway.
So we performed done some initial experiments to investigate mitophagy, and until now we couldn't see any difference in mitophagy in this model.
Okay.
And we are planning to do some proteome and transcriptome of this mouse model to have more insights about the mechanism and underlining of this alteration that is underlining these alterations.
Okay, thank you.
Thank you.
More questions? Let's gonna see here in the chat. Hi, Angela. It's great to see you. New results. Does UB2A mutants present other types of physiological change? Is there a specific UB2A target that might be responsible for strong phenotypes? Thank you. This is from Mariana Costa.
Okay, so Mariana, we don't know yet the specific target of UB2A. So for studying, it's Dimitris.
Dimitris.
So Dimitris,
Sorry,
we are going to do proteome for ubiquitination using brain of this mouse model. And we hope to find the targets of this UB2A in this model.
Okay, Jimmy. Angela, can you hear me?
I can hear.
Okay. More questions? Okay, Angela, thank you very much for a nice talk and a wonderful data. Thank you and congratulations. Now, my pleasure to invite Tanus Lima, who is a former student from my lab. Currently, he's working at EPFL in Lausanne, Switzerland, under supervision of Professor Johan Alkes. For those who doesn't know Alkes, I would say that he is the most important name is on the field of mitochondrial physiology, mitochondrial biochemistry in the world. And also it's important to highlight here that Tanus has great expertise in molecular biology of mitochondria. So he's gonna talk about sphingolipid metabolism and as a novel mediator of mitochondrial function and proteostasis. So Tanus, thanks a lot for coming and joining to us. Thank you.
Thank you, Leo. Let me share my screen first. Okay. Can you all see this?
Yeah, it's okay.
Is it working when I?
Yeah.
Okay, cool. Okay, so thanks, Leo, for the kind words, and thank you so much, everyone, for attending. Today, I'll talk about a little bit of our efforts to try to understand the role of ceramides in mitochondrial function and the cytosolic proteotoxicity that happens during the aging process, and with the focus more in the skeletal muscle. So as Leo mentioned, I'm working now at the Laboratory of Integrated Systems Physiology, coordinated by Johan Alkes in Switzerland, Lausanne. So this is our lab in this building of life sciences. And So Johan, he has many projects associated with the transomic population analysis, but he also have basic and translational projects in the lab in mitochondria and mitochondrial biology. And I started this work with him a while ago, I think 1 year ago. And let's start with the basis. So we all know that biological basis of aging is multifactorial. However, accumulated evidence among the 20 years from now have shown that mitochondrial dysfunction and the loss of protein homeostasis, which is called proteostasis, is one of the hallmarks of aging. And as you can see here, it's really remarkable how these pathways, such as protein folding and the mitochondrial pathways, are declines linearly across the mouse lifespan. One of the papers that we published a while ago show that there's a crosstalk between mitochondria function and protein aggregation or protein homeostasis. In this paper, we show that by boosting mitochondrial function with NAD, we can also improve the aggregation phenotypes of beta amyloid, specifically in the skeletal muscle. And the same thing that happens when we improve or we decrease protein aggregation in the muscle, we get a better phenotype in mitochondrial function. However, currently we don't know what signaling pathways are involved in this cross communication. So that was one of the questions of this work. And we thought that maybe ceramides could be a potential signaling interface in this cross communication because of this paper that was published in 2016, I think, by Andrew Dillon, showing that mitochondrial stress responses, or specifically mitochondrial to cytosolic stress response, is coordinated by lipid biosynthesis and specifically the ceramides. So when they knock out the molecular chaperone in the mitochondria, so increasing the protein aggregation in the mitochondria, this leads to a decrease in the ceramides levels, which is important for the expression of these chaperones and basically proteostasis targets, which will coordinate the proteostasis network in the cell, especially in the cytosol as well. So that was the first clue that maybe ceramides could be involved in this mitochondrial to cytosol proteostasis. So ceramides is a class of bioactive lipids, is part of membranes, but it's also signaling molecules involved in many cellular functions such as differentiation, proliferation, apoptosis, senescence, and also mitochondrial function. It's basically structured by a sphingosine backbone attached to a fatty acid molecule. So the ceramide can be synthesized by different pathways in the cell. The first one, the de novo synthesis here on the left, is happens on the ER, the endoplasmic reticulin, and it's basically condensation, started the condensation of serine and palmitic acid by the enzyme SBTLC1, and which by a series of chemical reactions will turn out to be ceramides. The other pathway is located in the lysosomes, is the recycling pathway, the salvage pathway, and the other one is the sphingomyelin hydrolysis, which happens in the plasma membranes. we're going to focus more on the de novo pathway because this is one of the pathways that is more associated with metabolism, since you have these substrates as the main drivers of ceramides. And there's many inhibitors such as myriocin for the first rate limiting step enzyme and other enzymes in the pathway available. So there's many studies using these inhibitors, and we also use them. So ceramides are critical in health, and when it's deregulated in several tissues, it has been associated with many pathologies such as insulin resistance, sarcopenia in skeletal muscle, chronic kidney disease in the kidney, in the liver, NASH, fatty liver disease in the heart, cardiomyopathies and arteriosclerosis, and in the brain as well, such as Alzheimer's and Parkinson. It's well known neurological diseases associated with increased level of ceramides. So I was, when I started this project, I wanted to combine all these concepts together, and I thought it was could be a good idea to check how ceramides are involved in mitochondrial homeostasis and also the cytosolic proteostasis during the aging process. But first, to have a little bit of clue if this could be a good project, we went for some publicly available data sets, which in several muscular diseases in humans, and we checked first the ceramide biosynthesis pathway. And we can see here that basically all these pathways are upregulated in muscle diseases, which we can see also the same thing for the proteotoxicity pathways. And it's inversely correlated with mitochondrial pathways. So this gave us the idea that maybe ceramides are upregulated that could be driving proteotoxicity, and this could be also associated with mitochondrial dysfunction in these muscle diseases. And if we look at aging, we can see here, and also the IBM, inclusion body myositis, which are conditions associated with proteostasis collapse. We see here in this data sets that ceramide biosynthesis is one of the top hits in rich pathways here. And also beta amyloid binding and agrosome formation as well. Which is, again, inversely correlated with mitochondrial pathways. So we started by looking at myoblasts, aged myoblasts, and also myoblasts from human IBM donors. And what we can see here in this pic here is that both old donors, myoblasts, and the IBM donors, myoblasts, they have huge aggregation going on the cells. So this red dye here is a proteostat dye, so it marks basically oligomers of the beta amyloid. And there's also increased mitochondrial function in the cells. So this is a good characterization of what we see before in the bioinformatics analysis. We are now analyzing sphingolipids in general, not only ceramides in myoblasts from these old patients, but we already had the sphingolipids measurements from the IBM donors cells. And we can see that many species of sphingolipids, including the ceramides in different chain lengths, are upregulated in these IBM donors. So we thought it could be, of course, a great idea to block the ceramides pathway to see if we could attenuate some of the effects of these diseases and also in aging. So we went for the most common inhibitor of the rate limiting step enzyme of the pathway. So we use myriocin for that. And we can see here in the cells that myriocin decreases robustly the ceramide levels in C2C12 cells and also in mice. So these are mice, old mice treated with myriocin for 5 months. As you can see here, there's a bit of increase comparing old versus young. So when these mice age, they have more ceramides, they have ceramide accumulation, which can be blocked by myriocin. So coming back to the cells, we treated the old donor myoblasts and with myriocin, and we can see here there's a major decrease in protein aggregation in these cells, and also we see a boost in mitochondrial respiration. So I also produced these cells. It's a C2C12 cell line, skeletal muscle cell line from mice, but I overexpressed the amyloid precursor protein with the Swedish mutation. So these cells, they are prone for aggregation. You can see here the red dye. It's really remarkable because they have the mutation, the Swedish mutation. So the beta amyloid, it's more cleaved and it aggregates more. In these cells, again, myriocin is able to decrease protein aggregation and increase mitochondrial function, as you can see in this seahorse experiment. And also by knocking down SBTLC1 in this APP Swedish myoblasts, we see the same effect as the pharmacological ablation of the enzyme, meaning that probably myriocin effect is not an off-target effect of the compound, but it's really associated with the decrease in the ceramide levels. In normal C2C12 cells, so we produce this SBT1 CRISPR knockout cell lines. We can see that it has a major effect on mitochondrial respiration in these cells when we knock out the SBTLC1 enzyme. And not only the function of the mitochondria, but also the mitochondrial transcriptional signature going from oxidative phosphorylation to unfolded protein response in the mitochondria and also mitophagy genes. We wanted to test this in vivo, but before going for the mice, we first used a worm model, and this is a cool worm model because it's almost the same thing as the APP cell. These worms, they overexpress the APP with the Swedish mutation, so they're prone for aggregation as well. They have beta amyloid proteotoxicity. In these worms, when we treat them with myriocin, we can see that the mobility gets better along the lifespan of the worm. This worm doesn't live long. They live probably 10 days max, and we can see that the mobility decreases along the lifespan, but myriocin is able to attenuate this decrease, which is also correlated with decreased paralysis and increasing lifespan. So myriocin also increased the lifespan of the worm. Same time, mitochondrial genes are upregulated. Mitochondrial function also is boosted by myriocin. We also checked the mitochondrial morphology network, and we can see that this, comparing this kind of Alzheimer model of worms with the wild type, we can see the loss of the mitochondrial network. But with myriocin, this Alzheimer worms, they are protected by this loss of the mitochondrial morphology. And also, these worms, they present a robust amount of aggregation, as you can see in the Western blots here. But with myriocin, we see a major decrease in protein aggregation, both pharmacologically with myriocin, also by inhibiting the enzymes, the SPTLC1 and also the SPTLC2 enzyme. And then we went for the mice. We treated old mice for 5 months with this compound, with myriocin, and so we blocked the ceramide production in the mice, or at least part of it, the de novo pathway. And we can see that this compound was able to increase mitochondrial respiration, especially complex 1, and in an oxidative phosphorylation state. Looking at these specific complexes, we can see in OXPHOS, we can see that complex 1 and complex 2 are the main affected positively by the compound, and not complex 4. And we can see the same thing that we saw in the cells and in the worms, that the transcriptional regulation of mitochondrial genes are also induced by the depletion of ceramide levels. And then, of course, we wanted to check protein aggregation in these mice. And for that, we did immunostaining with the A11 antibody that stains beta amyloid aggregation. And you can see that the old myriocin treated mice, they have less aggregation in the skeletal muscle. You can see here by the staining and also by this Western blot of 4G8 and by the dot blot of 4G8. So all of this impacted on the mice health span and phenotype, obviously. So they have a better running capacity, VO2 max. They have more strength, as you can see here in the grip strength. They have more coordination. We didn't check lifespan, but this data supports the idea that by depleting ceramides, you can improve health span in mice. So this is a really cool experiment. So I wanted to check what kind of proteins were aggregating during aging, and how could myriocin be affecting this? How myriocin could be beneficial in this context of protein aggregation? So what I did was a solubility fractionation. So I fractionate the proteins from the muscle as a soluble, this TBS, and insoluble fraction, the guanidine HCl. And I put this in a mass spec to see the proteins that were in the soluble and insoluble fraction in mice treated with myriocin or not treated. And we can see that most of the proteins that are being shifted in solubility are mitochondrial proteins. So what happens here is that when these mice age, they have less soluble mitochondrial proteins and more insoluble mitochondrial proteins, meaning that probably the mitochondrial proteins are being aggregated by aging. And the cool thing here is that myriocin was able to prevent this shifting of solubility of the mitochondrial proteins. We confirmed this also with the Western blot of the soluble fraction. So we can see here that specifically complex 1, complex 2, they are downregulated in the soluble fraction, in old mice. And Mariosin, which is this red bar here in the bottom, is able to attenuate this or even increase the amount of mitochondrial proteins in the soluble fraction. So that gives us the idea that the ceramide pathway is really involved with the mitochondrial proteostasis. So in summary, what I have so far is that ceramide depletion prevents the age-associated mitochondrial decline and reduces amyloid-like protein aggregation in human cells, worms, and mice. So this mechanism is conserved between species. At the organismal level, ceramide depletion was able to improve the muscle function and health span. And so we think that the ceramide biosynthesis wires the mitochondria to cytosolic proteotoxicity in aging. So this is the basic, what we think now. It's an ongoing work. We are doing a lot of other experiments to try to nail this mechanism, but we think that when these mice age, there's accumulation of lipids in the muscle. This is the main substrate for the enzyme SBTLC1 to produce ceramide, which might be acting on mitochondrial transcriptional regulation and also the cytosolic aggregation, such as the beta amyloid aggregation. And also this is also impairing mitochondrial proteostasis. And in this work, we see a higher or a stronger effect on the complex 1 of the mitochondria. So this creates kind of a vicious cycle, because you have dysfunctional mitochondria will accumulate more lipids and so on. And Mariosin, by blocking the SBT1, is able to attenuate this vicious cycle of ceramide muscle aging. So I think this could be a good target for muscle diseases, for even myopathies or desminopathies, or even muscle dystrophies as well. So thanks a lot. That's all I have so far. Let me thank here first Johan Auerks, who's supporting me here in Lausanne. Really nice to work with him. And some of the people in the lab, so Pirko Perka, he's not in the lab anymore, but he did a really nice work in the beginning with the ceramides, Martin, Mario, Hao with the bioinformatics, John David with the help with the proteomics, Juliana with the lipidomics, and of course Leonardo Silveira with all the support from Brazil. And I started this project in my FeSBE, so I need to thank a lot FeSBE and the EPFL for now paying my salary. So thanks a lot. That's it. Thank you.
Thanks a lot, Tanus. Very warm find and very clear presentation. Now we are open for questions.
We have one on the chat now.
Okay. This is from Dimitrios. Hi, Tanus, very interesting results. I'm wondering if there is a specific pathway activated by ceramides, protein kinase C, for example, that could help explain that reduction in the mitochondrial function.
Yeah, actually we see an increase in mitochondrial function with the decrease of ceramides. I didn't play a lot with increasing ceramide levels and checking the pathways, but from the proteomics analysis and the pathway analysis, one of the things that stood out was translation in general. So basically what happens is that all global translation is decreased. So we think that maybe this is a signature of stress responses. It could be maybe the integrated stress response by EIF2 alpha. I'm checking that as we speak. Don't have much data yet, but yeah, but you know, for example, if you think about ceramide as a signal of nutrient status. So if you ingest a lot of lipids, you have more ceramides, right? And if you do caloric restriction, for example, you probably have less. And this is one of the pathways that are associated with lifespan during caloric restriction or fasting. Are the decrease in mTOR pathway and increase in AMPK pathway. That could lead to more mitochondria, more fatty acid oxidation in this condition. So maybe, I don't know, maybe the ceramide could be a signaling interface also in nutrient status. And actually that was already proposed by Scott Summer. But one of the pathways, one of the effects of this caloric restriction, for example, is the downregulation of the translation. So maybe downregulating translation could be something good for the proteostasis network, because you have less burden of protein aggregation. So I don't want to, in this work, I don't know if I really want to explore specific pathways, but I'm more into trying to check everything and come up with a more general, broader idea.
Okay, you have another question here. It's from Carlos. Great talk, Tanus. How ceramides would affect gene transcription? Any previous evidence?
Sorry?
How ceramides would affect gene transcription? There is an evidence.
Oh, okay. Okay, yeah, that's a good point. Let me think. Well, if we think about this AMPK pathway, that could be one of them, right? So you have more AMPK when you block the ceramide pathway, then you get all the downstream transcriptional regulation of this pathway, which is known to be mitochondrial associated. We did some RNA-seq. I did some RNA-seq from the mice, from the cells. Not much going on there. We don't see, a huge effect on transcription. So that's why we think that maybe it's more associated with translation and proteins, but we are not that sure. One of the problems that I think I might have, and we're already working on it, is that we use myriocin. As you know, myriocin could have off-target effects in other tissues and everything. So we are now transducing mice with AAV directly in the gastrocnemius to knock down the SBTLC. If this thing is really muscle specific and SPT1 is specific and not an off-target effect of the compound. I'm doing this experiment now. Mitochondrial function worked great. We have higher mitochondrial respiration in this muscle. I'm gonna do RNA-seq and proteomics to check the pathways. Maybe that will give us something that we didn't see in the myriocin treated mice.
Okay, anybody else see more questions? Tanus, How did you see the effect of myrcene on basal respiration in cell experiments? You did not see effect on basal respiration, but you really did it during maximal respiration.
Well,
I mean, maximal respiration is like a spare capacity, you know? And, sorry. Pessoal, acho que tem algum microfone aberto, por favor.
Primeira pergunta que eu fiz.
Yeah.
Okay, spare capacity is expected to be used when you make exercise, for example, when you increase the rate of oxygen consumption or something like that, you know? So how did you see these finds on basal respiration? Sorry.
Yeah, for me, it could be related to fatty acid oxidation. So maybe the complex 2 is in the cells, right? Because I checked in the mice, we have a more pronounced effect on complex 1. But the cells, I didn't perform the complex respiration, but it could be associated with the succinate dehydrogenase with a more fatty acid oxidation in the cells. That I would think that, if you see different systems, you have different readouts. With the old donor myoblast, we see something here in basal, but the APP cells, we do only see the maximum capacity and spare capacity. And then in the mice, it's a little bit different. When we do genetic ablation, we see a huge increase in all of them. So it's hard to know what I think it's just boosting all the mitochondrial homeostasis. It's probably not only mitochondrial biogenesis, I guess. I would suppose that this is also associated with a better mitochondrial stress responses. UPRMT might be involved. And also mitophagy. I have some data with mitophagy showing that ceramides are involved in mitophagy. So that could be also something to tackle.
Okay. So thanks a lot, Tanus. Wonderful presentation and finds. Thank you.
Thanks.
So guys, now we're gonna make a 5-minute break, and then you come back with Professor João Paulo Campores. Thank you. Ei, Ana, tudo mais calmo aí agora?
Tudo. Eu dou um informe sobre o congresso, então, depois do break.
Desculpa, você ia falar agora, né?